right. Some proteins have prosthetic groups attached to them. The term is a prosthetic group. And those proteins are called conjugated proteins. If a protein has a prosthetic group attached, that means it has a non-protein portion to it. So it could be a lipid, or it could be a carbohydrate, or it could be a metal. Those are conjugated proteins. So a lipoprotein is a protein that has lipids attached to it. A glycoprotein is a protein that has carbohydrate attached to it. A metalloprotein is one that has a metal, like maybe a magnesium or a zinc attached to it. A nucleoprotein has a piece of nucleic acid to it. So all those are in a big category called conjugated proteins. If a protein doesn't have a prosthetic group attached to it, it's just a simple protein. Okay? The last thing on our what you need to know list is reactions of proteins. There are two main things that proteins will do. You can hydrolyze them or you can denature them. Let's first talk about hydrolysis. Hydrolysis, as we saw in previous chapters, is we're going to break the peptide bonds. Remember, hydrolysis of carbohydrates, we broke the glycosidic bonds. Hydrolysis of the triglycerides, we broke the ester bonds. So when we're hydrolyzing a protein, we are breaking the peptide bonds, and you end up with free amino acids or just smaller peptides. How do you hydrolyze a protein? You can either use an enzyme, and that's what happens in digestion in your body. You've got enzymes that will hydrolyze your proteins, break them up into free amino acids or smaller peptides, or you could use acid and heat. Okay, so if you've got, let's say this alanine, tyrosine, threonine, lysine, aspartic acid, and you are going to hydrolyze it. So whether you use an enzyme or an acid or heat, you could chop it up into individual amino acids or chop it up into smaller peptides. So for example, you could maybe chop it up right here. And then you would have an alanine tyrosine dipeptide, and you'd have a threonine lysine aspartic acid tripeptide. So it depends on where the enzyme is going to break peptide bonds. Some enzymes are well known that they cut in certain locations. Other enzymes, hydrolyze in other locations. Some just look at and find a specific, a specific amino acid, and they'll cut right after that amino acid. If you're just boiling it and you've got acid and heat, it's just a random bunch of breaks, and you end up with usually just a whole bunch of free amino acids. But like I said, you can break it into smaller peptides. So if, let's say, you had a protein that was 50 amino acids long and you started to hydrolyze it, you could get several smaller peptides, dipeptides, tripeptides, pentapeptides, and then you could further hydrolyze it to make it all free amino acids. When you've got all the free amino acids, that's when your digestion is complete, and now they go through the villi of the small intestine into the bloodstream. All right, denaturation is different. Denaturation, you do not break any covalent bonds. Now, all these peptide bonds are covalent bonds. So when you denature a protein, you do not break any peptide bonds at all. But you do break hydrogen bonds, hydrophobic attractions, ionic bonds. So all that, the three-dimensional structure, so quaternary structure gets messed up, tertiary structure gets messed up, secondary structure gets messed up, primary structure doesn't, not with denaturation. So denaturation basically unravels the protein, and it basically alters the 3D structure. When you denature a protein, you usually have messed up its function, okay? If a protein needs to be folded in a specific way in order for it to be functional, in order for it to do what it's supposed to do, and you denature it, you open it up, and now you've got all these amino acids. They're still in the same order, but they're not folded correctly. And remember, that folding is really what gives that final 3D shape that makes it a functional protein. How are you going to denature a protein? You can apply heat. That will denature it. Remember, hydrolysis is heat and acid together, but heat alone is going to increase the kinetic energy. It's going to mess up all the hydrogen bonds and all of those, skeletal hydrogen bonds between the carbonyl and the amine groups, all the side chain hydrogen bonds between all of the polar neutral groups, they all start to break. So you've altered the structure. Change the pH. Remember, if you're going to alter the pH, you're going to alter the charges on the side chains. And sometimes those side chains have to be at a specific pH in order to create that specific bond. Remember the example I gave you guys, lysine at a pH of 7 is positive, aspartic acid at a pH of 7 is negative. Well, if you take that protein and you throw it into an acidic medium, now the lysine is still positive, but the aspartic acid doesn't have a charge anymore. Remember, it gets protonated. So you've just broken the ionic bond. So if you mess with the pH, because every protein has an optimum pH and it only works at that specific pH with a little bit of variability. So you alter the pH, you alter the charges on the side chains, it's going to denature the protein, it starts to unravel, you end up with a non-functional protein. Another thing that could denature a protein is heavy metals. What do I mean by heavy metals? I mean heavy molecular weight metals. For example, mercury or lead. Mercury and lead will denature proteins. They will come in and they will interfere with proper ionic bonding. For example, if you have an aspartic acid that's normally ionically bonding with a lysine, for example, and then you throw in a lead ion there. Well, now the aspartic that's negatively charged starts getting attracted to this lead ion, which is with a +2, and maybe two aspartics start to get attracted to it instead of doing their normal ionic bonding. So you throw in heavy metals, you're going to denature a protein. And then finally, the last thing on my list that will denature a protein is alcohol. Alcohol will also disrupt hydrogen bonding. So you get disruption of hydrogen bonding with heat and alcohol, disruption of ionic bonding when you alter the pH and with the heavy metals. Alcohol has that OH. If you've got an ethanol there, it's got that OH, and that OH can potentially come in and interfere and hydrogen bond with the side chains and mess up the 3D structure of the protein. What happens when a protein gets denatured by these things? Well, the solubility goes down. Solubility goes down. If you have an egg white, which is all protein, albumin, and you heat it up, what happens to that egg white? It could be soluble in water before you heat it up, but the minute you heat it up, you turn that liquidy egg white into this solid, hard-boiled white egg. That's not going to be soluble in water. All right, so solubility goes down whenever you're denaturing a protein. And think of it as, remember how a protein folds and hides all those non-polar side chains in the interior? Well, once you denature a protein, the protein starts to unfold, open up, all those hydrophobic side chains, all those non-polar ones start to go out to the outside. And then there's water, and they're not soluble in water. They're not attracted to water. They're very hydrophobic. and so the whole protein goes out of solution. You tend to see that in lab, and you'll be able to witness the whole denaturing process and the decreased solubility. Other things happen with denaturation. Basically, usually we'll have a loss of function. So, for example, if it's an enzyme and that enzyme has to catalyze a specific reaction and you've denatured the enzyme won't work anymore. It will not catalyze the specific reaction. Okay? All right, and that pretty much does it for the protein chapter. Make sure you read your book, get all the extra information from the textbook, and I will see you in the next video.